Hello, folks, and welcome to Lesson 5 of Unit 1 of the Higher Human Biology course. Today, we're going to be covering human genomics in this lesson. Now, this is one of the shortest of the topics in the course, so it won't take us as long as our other lessons. However, there are some important things here that we make sure that we learn just in case it obviously comes up in the exam. And we'll do a couple of exam questions at the end as usual. However, again, there are not many questions from the past papers because it's such a small topic. So we'll do our best. We'll go through them all and we'll obviously go over everything. But just keep that in mind. It's not the most assessed part of the course. But nevertheless, we should still treat it with equal effort and learn all the content just as well. So let's talk about sequencing DNA. So when we talk about human genomics, and there's a very, probably it seems very old to you guys, but happened in my youth. There's a very old project called the Human Genome Project, and that's where they between 1990 and 2003, so 13 years they spent on this, and they spent over $3 billion on it. And it was a cross-country effort. Actually, it's had the scientific collaboration that we don't quite see as much these days in the modern world. But all the major countries and so on combined together to work on this. And what they managed to do is they managed to sequence the entire 3 billion base pairs that we find within the human genome, which is quite a remarkable feat. And as I say, at the time, it took 13 years and $3 billion. What's interesting is, like almost all technology, both the cost and the time to do this has come down dramatically in recent years. And only just a couple of years ago, I got my own genome mapped, my own human genome mapped, and it cost, I think it was £90, so less than £100, and it came back within a couple of weeks. And it's the speed at which technology goes and the ability, obviously, of like things like computers and other programs as well to help with these things mean that tasks which once took us 13 years and $3 billion are now taking us a couple of weeks and £100 from, a local lab. So, but sequencing DNA, it was once thought of as a huge challenge, and obviously now is something which is very much achievable for us, and we're able to do quite quickly. So the genome of an organism, I've mentioned the word genome a few times, and so let's talk about what it is. The genome of an organism is its entire hereditary information encoded in the DNA. That's a bit of a mouthful, that first sentence. I'd still try and memorize it as it is for the exam, but what that means in sort of normal terms is the genome of an organism is all of the genetic information that you have inherited from your two parents, and that's encoded in our DNA. Okay, so hereditary information. Now, the genome is made up of both genes, as the name suggests, genome is made up of genes, but actually is made up of a lot more other sequences that do not code for proteins. So we know that genes are the codes for proteins. We know that from the gene expression topic, but only about 1 to 2%, I think it's about 1.5 to 2% of our genome is actually genes. The rest of it is other sequences which help in things like gene expression and so on, and have lots of other tasks rather than just being genes that make proteins. So that's worth remembering as well. The genome is made up of genes, but there are lots of other DNA sequences that do not code for genes. So we talked about sequencing DNA, and the Human Genome Project was able to sequence the entirety of our DNA. So what is genomic sequencing? Well, in genomic sequencing, the sequence of nucleotide bases can be determined for both individual genes and the entire genome. So, for example, they could do genomic sequencing on your DNA and, in theory, could give you the full list of the 3 billion base pairs that exist within your DNA in the correct order as well. And you can see a wee image here at the top. That's a tiny fraction of what the genome would look like, but you can see how basically unreadable that is to most humans, right? If you looked at that, you wouldn't know where to start. But fortunately, there are people with a trained eye who do know where to look, and they've obviously worked on this for a long time. But more and more recently, the use of computer programs have been used to help scientists to identify base sequences by looking for some of the sequences that are similar to the genes that we already know about. For example, if you're looking for a gene through a genome for a particular condition, an inherited condition, and you know that from other patients who have the condition, they have a certain sequence on their DNA, then you can look for that within the person you're testing. Now, For a human to do that, to read someone's 3 billion base pairs and to find a sequence, or even if even if you're only looking at a small section of the DNA, even still, it's going to be overwhelming. So what they do is they use computers to help them with that, and they can obviously do that much faster and usually with fewer errors and more accuracy than a human might do. Now, the use of computers doesn't just stop there. When we are comparing sequence data from, say, a human and a sample found at a crime scene, or perhaps we are looking at how related two people are to one another or something similar, we use both computer and statistical analyses, which collectively are known as bioinformatics. And there's a little image here that just shows you that crossover between biology, statistics, and computers there, which all merge together to form that field of bioinformatics. So, yeah, when we're comparing sequence data, we have to use our biology knowledge, but we also have to use our statistical knowledge as well as our computer science ability as well. The use of computers and all of the programs and software and so on that are available now on computers, that is called bioinformatics, and that's a word that's very much worth remembering. Bioinformatics. So we use computer programs to identify those base sequences, and in genomic sequencing, we can determine the entire sequence for both individual genes and the entire genomes of people. The genome itself is made up of genes, but also other DNA sequences that do not code for proteins. And our genome is the entire hereditary information found within our DNA. Okay, so hopefully that first bit all makes sense. I get most of this stuff is just it's just about learning it. There's not likely to be any great deep questions in the exam on this topic, or certainly there hasn't been in the past. Most of the questions are just a multiple choice question, and we'll look at a couple of those at the end just so you can see how you're applying this knowledge. So the second part of this topic talks about the uses of these genomes. When we sequence someone's genome, what can we actually use that information for? Well, an individual's genome can be analysed, and it helps us to predict the likelihood of developing certain diseases. Because what they can do, as I said a moment ago, is they can look for known sequences. That's sequences they already know about that they can attribute to a particular disease. And if you have that sequence within your genome, then they know that you are likely to develop that disease. And we can use pharmacogenetics, which is the use of genome information in the choice of drugs. What they can do is they can use an individual's personal genome sequence, and they can use that to then select the most effective drugs at the most effective dosage to treat their particular disease. And we call this personalized medicine, okay, where we use pharmacogenetics. That's a study of drugs and genetics together. Pharmaco is drugs, and genetics obviously is our genome. And we can then make a good choice in terms of the personalized drug that we give to that person, and it will be at the most effective. This is the key word. At the most effective dosage, as well as choosing the most effective drug for that person. And that is an example of personalized medicine. I think for all you young people that are studying the higher course just now, you will probably experience this in your lifetime, where you will be getting not just the generic antibiotics for a chest infection, but you'll be getting a specific drug at the particular dosage that is going to best treat your chest infection. Okay, so they'll be looking at your personal genome sequence to help them with that. And this study of drugs and genetics and the relationship between them is called pharmacogenetics, and it helps in terms of the choice of drugs for that particular person. We can also look at the person's genome as well to analyze it to predict the likelihood of developing certain diseases. Okay, so that's the human genomics topic, and that is the main things that you guys need to know about for your exam. Now, as I say, it's not used in great depth in the exam in previous years. That's not to say it won't come up this year, and you have to know all of this stuff. But I'm going to show you a couple of questions that have come up in the last few years in the multiple choice section, and obviously to go about answering them. So I hope the lesson part's been useful, a nice recap over those parts of the human genomics topic. Let me know if there's anything you struggled with or you still have questions about, and I'll see you in part 2. Hi folks, welcome to the past paper section of the human genomics topic. As you can see here, we've got 3 multiple choice questions which I've managed to source from past paper questions. They're a bit scarce, in all honesty, the questions on this topic. There's not too many of them, but we will have a look through the ones that there are and talk about how we would answer the questions. So as usual, you can pause the video if you want to answer it before I steam ahead and give you the answer and explanation. Okay, bioinformatics is the study of information from, so we've got a choice here, DNA transcription, not going to be that one. DNA sequencing, that sounds right for this topic. Nerve impulses in the brain and receptors of nerve endings in that. So it's going to be B there, for that one. Pharmacogenetics, so pharmacy, drugs, genetics, or genome, is the use of gene sequences to assess the likelihood of passing on a disease. So this bit sounds right, the gene part, but the passing on of a disease, I think that's not going to be one of these, okay? That's not going to be one of the uses of pharmacogenetics. Computer programs, well, we know computer programs are involved in bioinformatics, so that's not going to be right. And similarly, we can eliminate D because it talks about computer programs as well, which we know is bioinformatics. So we look at C, we've got gene, okay, genetics, pharmacogenetics, and that's to select appropriate drug and dosage to treat a disease. So pharmaco, drug, genetics, gene, drug. So the answer is going to be C for that one. What term describes the use of genome information in the choice of drugs that may be prescribed to a patient? Okay, so genome, gene, and choice of drugs. We know that that's going to be pharmaco, drugs, genetics, genes. Okay, so that goes with that, and that goes with that. Okay, so the word itself is, we can look at the meaning of it and we can see from the question the aspects of that word, genome and drugs, pharmaco and genetics. Okay, and so it's going to be C for that one. Now, I hope those questions have helped. As I say, there's not been many of them in the exam papers, so not a lot to go off there, but if you get a question like that, hopefully you understand it. But I'd say the key things are know what bioinformatics are, know what pharmacogenetics is, know what personalized medicine might involve, and know how we sequence genes through genomic sequencing, and obviously what our genome is composed of. If you can do all of those things, then I think you're going to be pretty good with whatever they throw at you in the exam. Okay, folks, so thanks for watching the video. Let me know in the comments if you've got anything else to add or you want to ask about, and I'll see you all next time for the next lesson. Class dismissed.